Hi, Benjamin here, and welcome to Breakthrough Science. I have the pleasure to be here today with Dr. Lynn Rothschild, who is actually an astrobiologist at NASA Ames Research Center. She's furthermore an adjunct professor at Brown University and University of California, working on the boundaries of life, synthetic biology, and much more. Dr. Rothschild contributed over 90 peer-reviewed publications to the scientific community and has contributed to space missions and is proudly hosting the iGEM team from Stanford and Brown University for several years. Furthermore, she's currently also supervising my research, so it's even more pleasure to be here with her today. And with all that said, welcome.
Thanks so much, Benjamin. I'm just thrilled that you're doing this series.
Great, thank you. And let's start to dive into the first question. So, many of my listeners asking themselves, how did life on Earth emerge? And how did this process came together? And what is your opinion? How did it start?
Well, I wish I knew the answer. And in some ways, this is life's greatest mystery. The funny thing is, if you go out into the public, a lot of people think the great mystery is going from a bacterium to a human being. But in fact, for a scientist, the biggest mystery is going from a rock, something that's not living, to something that we would consider alive. And you would think that that would be so simple, but that is really an incredibly complex answer. Well, maybe it isn't complex. Maybe there's just some trick that we don't know. So a lot of people are working on trying to figure out what the environmental conditions were like, what are plausible scenarios to go for the origin of life. But even if we do all that and recreate life in the lab, that doesn't mean that that's actually the way it happened on the Earth. So some of my interests have been, what are the possible building blocks of life? What does nature tell us about what's possible? And go from there.
So starting with a synthetic cell or regenerating a synthetic cell in this direction, or more like really figuring out the single building blocks?
Well, a little of each. Those are good points. So you haven't brought it up, but I will bring up the whole question of an RNA world. Now, this has been a very popular idea for the origin of life for literally decades. And the whole idea is that RNA, that nucleic acid that really interfaces between DNA and proteins in modern cells, could have been the original molecule of life. And the reason that this is such an appealing idea is that RNA can act as genetic material, it can preserve the information, but it can also have some catalytic activity just like a protein could. So you think, here's one molecule that can do everything. So a lot of people have tried to start with this one molecule and build life. And so here's an example where I got into the field just really a few years ago, although I've been in astrobiology, microbiology, and so on for many years, and asked myself as an evolutionary biologist, well, what would be the environmental conditions? They wouldn't be a test tube of things you bought from Sigma. They would be some kind of environmental conditions. And if you start to look at what is in meteorites, in comets, in the sort of simulations that people do in the lab, like the sort that Urey and Miller pioneered really years ago, what you don't see here is RNA. What you see, for example, are amino acids, which can easily be made into proteins. And so that's what I mean by an environmental condition. RNA is a terrific idea. It's extremely appealing because it can do all these things. But if it's not there, it doesn't matter how good an idea it is. It's not likely to happen. So therefore, our research has really started to look at things like proteins and what function they could have had in the origin of life.
Great. So that's a very interesting, because just one video before I had an interview with Mark Ditzler, who is more on the RNA world side. So we see a little bit of different information, but also information coming together. So, and you actually directly took away the next question, which is, how is your research related to it? So it's mainly the...
Well, and don't get me wrong, because I think that RNA was probably very important. What's kind of interesting is that there are all sorts of molecular fossils in the cell. It's almost like in evolutionary biology is like a crime scene. You have to be a detective and go back and say, all right, here's the empty room and a smoking gun on the floor, whatever, what happened? And we're sort of that way with evolutionary biology. We say, okay, here are modern cells that are functioning. How did we get here? And the smoking gun for RNA is the fact that we use RNA in a whole lot of different ways in modern cells. Not just for messenger RNA for transfer of information from the DNA, which encodes it to make proteins, but also ribosomal RNA. You get transfer RNA. We now realize there's silencing RNA and there are aptamers, and it goes on and on and on. So it looks like there are all sorts of tantalizing clues that RNA could have been, the big gorilla at one point in the origin of life. And that I do buy into, because there are too many hints that are left in the cell. But to think that life started with a single RNA molecule that somehow accumulated all this stuff around it, I think is kind of crazy. So you asked me what my idea of the origin of life is, and maybe you'll be very disappointed in me. But I don't think it was a very clean single molecule in a test tube, because if you go out into nature, you don't see single molecules in test tubes. What you see is a lot of garbage, for want of a better word. You see mixtures of compounds. And what must have happened, I believe, is you have different mixtures that may have been encapsulated into small units, into like little cells with lipids around, which we know can be made abiotically. So think of it as sort of like single cell encapsulation. And my guess is that most of these didn't have a single molecule. They probably had mixtures. And some of those mixtures work better than others. It's just like, what a terrarium is, where you put, like maybe snails and algae and so on, or plants in a single enclosed system, and you expect it to work. Well, if you put in 3 snails and no plants, it's going to die. If you put in 4 plants but no snails, you're going to have a different scenario. And I really believe that the origin of life was that sort of thing. It was like a terrarium. If you had a combination that worked, you could take off. But most combinations probably just ran into the ground.
Terrific. So, Detective Rothschild, then, for all the options which are out there, which one is your personal favorite? Do you think that the small terrariums were most likely in a favorable condition at the hydrothermal vent, or on another planet and just were transported with a meteorite? Or do you think it all happened in a small volcanic pond somewhere on the surface of Earth?
Well, I have to say that I think it probably happened on the Earth. It could certainly have happened elsewhere too, because we think it's a physical chemical process, and we know it's possible. Whether it's likely or not is another matter, but we know it was at least possible. So we leave that aside for a moment. You asked about hydrothermal vents, and I will give you the same answer I give everyone, that I know a lot of chemists who are far better at chemistry than I will ever dream about being, who believe passionately that it had to be in a hydrothermal vent. And I know other chemists who are equally good who say it's absolutely impossible. But I will tell you what I do think is a critical thing is the problem of dilution. And that's something that my great hero Charles Darwin brought up too. If you make the perfect RNA molecule or some proteins or whatever in the middle of a pond, not even getting to an ocean, they're going to dilute out, and so you'll never be able to have them find each other to actually go to making a cell. And so you do have this dilution problem. You need to have a situation where you can concentrate the molecules. My guess is you're talking about a mineral surface, but another possibility is wetting and drying. For example, our good colleague at University of California, Santa Cruz, Dave Deemer, who's thought greatly about this and done many experiments over a very lifetime-long career, has an idea that life could have originated in the splash zone around geysers, like places like Yellowstone National Park. Pictures of that behind me. So you imagine you've got splashing, and maybe it's hot, maybe it's not, but you've got splashing onto rocks around you, and then that material evaporates, the water evaporates, and so you have a concentration, and then you've got more splashing, so you get a good concentration of molecules. And so I believe that kind of thing, the importance of having a concentration, whether it happens in a small sort of hole in a hydrothermal vent, or whether it's because of a rock water interface in an ocean or a pond, or whether it's drying in a splash zone, I don't know. But I do believe that that is actually incredibly important.
So the spatial boundaries and
That's
the
right.
drying. And,
You have to
wet
solve the
places.
problem of dilution.
Yeah. Did the understanding for you personally change via any publication or any happening in in recent years? So the way you understood it, or the way you saw the origin of life, did it fundamentally change during your career or in the last few years?
Ooh, I think for me, really, the issue was the one I mentioned before, and that is what was actually there, because I was very happy to go along with the RNA world until I started to look at what was there. I just imagined that there were ribonucleotides all over. Now, they have found bases in meteorites and so on, but that's different from talking about a full-on ribonucleotide. But again, you do find amino acids all over. So I guess one of the papers that really has influenced me over the last few years is one by Paul Higgs and Ralph Pedretz, wonderful scientists up at McMaster College in Canada. And what they did was look at what the amount of amino acids were in these various prebiotic scenarios. So looking in the Urey Miller sort of experiments, and in meteorites and comets and interstellar dust particles, I think, and so on. So they took the amino acids that are found in our bodies and all of life on Earth, and put them in an order that you find them prebiotically. And that I thought was absolutely fascinating, because this is a clue that these would have been the amino acids that would have been used first, because if there's 10 times more glycine, say, than serine, you would think that the early proteins would be primarily glycine. Alanine's up there very high. And in fact, there are really only 4 or so amino acids that are very high up there, and maybe another 6 that you find. Now, what's really interesting is about half of the amino acids that we use are not found in abiotic settings at all. So these must have been the ones that were built by life. So that, to me, I don't know, it tripped a switch. And I end up showing that as one of the figures from that paper all the time, that again, life must have used what's out there. It's not just that it's a good idea, but this item has to also be available off the shelf. And so I think that really, it's influenced some of what we've done with our look at the evolution of cysteine, for example. And it's kind of a funny thing, because I think that in school, of course, you're taught about evolution, which parallel would be history. But for some reason, people think of physics and certainly chemistry as immovable, or even, oddly enough, sort of origin of life, that, oh yeah, you always had to use 20 amino acids, or in the case of chemistry, periodic table is always there. Well, it wasn't. What happened is, at the Big Bang, you got the production within minutes of hydrogen, helium, and a little bit of lithium. Everything else had to be made much later, and so even the periodic table had an evolutionary history to it, and certainly the components of life have as well. And so that's something that really I've been very interested in over the years.
So it would be a good idea to make another session about the origin of chemistry.
It would be. That'd be fun.
All right. So that also, I guess, was kind of the most shocking, because that would be another question. What was the most shocking finding for you, or something which changed your mind? But that's basically going hand in hand with that.
Well, let me say there is one other article, and I keep wondering whether you were thinking of this one when you asked the question, and that is John Sutherland, who is at the University of Cambridge and is a very fine chemist. Had some papers a few years ago that really did surprise much of the community, where he was able to make RNA in the lab in a way that was totally different from what people had assumed was the way you made it. And there was some attempt to use natural settings, but I do always wonder, and again, I'm not a great chemist, is how realistic are those series of reactions out in nature? I'm still not sure that they are. And there have been other really wonderful papers over the years. I love the work of Peter Pinheiro. Also, at the time, he was a postdoc at Cambridge with Phil Holliger. He now has his own lab at the University College London, where they started to look at xenonucleic acids, XNAs, which I love because that sounds very NASA, alien nucleic acids. But there's no reason you would have had to use adenine and guanine and cytosine and thymine and so on. Why not some of these bizarre ones? And I love talking to people like Vitor because I say, I want an organism that can withstand higher levels of radiation for either the early Earth or life on another planet. And I said, what can you do for me? And he'll look at DNA and say, well, if we could get rid of this double bond and we could add something here and so on, I think you'll be in better shape. I mean, I just have enormous admiration for chemists who can do that sort of thing. But it does make you wonder, is there more than one way to have life originate? I just think of the expression, I guess maybe it's an American expression, is there more than one way to skin a cat?
That's great. It's very interesting also to hear that there are ways we don't even, or we could not even imagine yet, or some people do, which could be the origin of life. So, yeah.
Absolutely, and I used to tell my students that right now we don't know how life arose. We don't even know how long it took. People say, it had to have taken a long time, so therefore you need to have a planet around a star that's small enough to last for billions of years. Well, that's silly, because we have indications that there could have been the origin of life within 100 million, 200 million years of the Earth cooling down. And you think, well, that's really short. And you think, well, but 200 million years, that's really long. Well, the fossil record's lousy back then. How would you even know? That's a maximum time. Maybe it's one of those things that you have all these like little reaction vesicles out in nature, and one of them takes off one day. Maybe the origin of life, once you have enough variety out there, is a 3-day weekend. Maybe it's a bank holiday, and maybe it really is 100 million years. I suspect that it's a long time to start to get enough building blocks of the right sort of things and get the right combination. And once you start to get combinations that work and they mix and match and so on, it could be very fast. And so I used to tell my students that, maybe today it's the mystery of mysteries, and maybe in 20 years there'll be some trick that we've learned about, and I'll pass out test tubes on Friday and say, go make life. I'll be collecting these Tuesday, and whoever has the most creative life form gets an A.
That sounds like a terrific idea for a student project. I would be totally in for that.
I'll sign you up.
So with all that said, thanks a lot for this great interview. It was
My
a pleasure to talk to you.
My pleasure.
And thank you all for listening to this interview, and see you for the next time. Bye bye.